Welcome to my developmental biology playlist. In this video, we would look at the first week of embryonic development. So stay tuned till the end of this video and don't forget to subscribe. Let us look at the female reproductive system to understand the context of this process. In the female reproductive system, there are two ovaries on the two sides, and these ovaries produce ovum or the female gamete. And this process is known as ovulation. In a separate video, I have described in details what is ovulation. So click on the video to understand more. Once ovulation happened, the ovum would try to migrate through the fallopian tube and it would reach the uterine cavity. Now imagine at the same time there was sperm present in the female reproductive tract. So it would try to fertilize this ovum. It turns out that there are 200 million sperms that is ejaculated in one round of ejaculation, but only 200 of them survives and make it to the oocyte. Out of these 200, only one would fertilize the ovum, and after the successful penetration, it would ensure other sperms cannot penetrate the ovum, and thus it prevents polyspermy. There would be a separate video on polyspermy as well. All the links are in the I button. Now this penetration process is not easy because there is a thick coating of granulosa cells known as corona radiata. Sperm has to penetrate its way through the corona radiata to reach the nucleus. Then it has to penetrate zona pellucida. Once it has penetrated all these layers, it would eventually inject its genetic material in the cytoplasm of the ovum. Then the male and the female pronucleus would fuse together to form the zygote. Now, after the formation of zygote, if we wait for 30 hours, the cells would divide. A cleavage furrow would run through the entirety of the cell and dividing it into two individual cells. This is the process of cleavage, and it results in formation of two cell stage, and this is happening at about 30 hours after the zygote formation. If we wait 60 hours, there would be 4 cell stage, 8 cell stage, and eventually after the day 3 to 4 there would be 16 cell stage, and at day 4 to day 5 there would be a 32 cell stage. One of the noticing factor in this particular process is all these events are happening while the embryo is moving towards the uterine cavity. Eventually in the uterine cavity, the embryo gets embedded into the uterine wall. Now while these divisions are happening, one important thing that we should understand, the cell number is increasing, but the cell size doesn't change that much. Even after the end of 8 cell stage, the cell size overall remains the same. Now at the 16 cell stage, which is known as morula because of its mulberry-like appearance, we can see the zona pellucida still covers the overall cells. It's like a bag that covers these blastomeres. After 16 cell stage, there would be 32 cell stage, and this structure is known as blastocyst, and this structure would be readily implanted in the uterine membrane or in the uterine wall. So blastocyst would shed its zona pellucida and get implanted into the uterine cavity. This would be the initiation of the second week of embryonic development, and this implantation takes place in day 7. If you want to learn more about the events of second week of development, click on the video in the I button right now. All the flashcards associated with these topics can be found in my community post. Don't forget to like, share and subscribe. If you wish to follow me on social media, you can, and please get connected. Do let me know your suggestions in the comment. Thank you.